Hello friends, welcome to AgriGPV YouTube channel. In this video lecture, we are going to discuss a very important aspect in quantitative genetics or plant breeding, that is breeder's equation. So what is breeder's equation? Actually in plant breeding or quantitative genetics, we use a equation that is R equal to H into K. This is known as breeder's equation. So what is R? R is response to selection. R is response to selection or genetic gain or genetic gain, while H equal to heritability in narrow sense. Heritability in narrow sense. And here the K is selection differential. Selection differential. In our previous video, we have discussed genetic gain or response to selection. You can watch the video. The genetic gain is the improvement in the mean phenotypic value of progeny of selected population over the progeny of unselected population or base population. Okay, so this is the genetic gain, and genetic gain will enhance if the heritability of particular character will enhance, and when the selection differential will enhance. What is selection differential? Selection differential is the difference between phenotypic value of selected population and base population or unselected population. Selection differential is the difference between mean phenotypic value of selected population and mean phenotypic value of base population or unselected population. So if the heritability narrow sense will increase and the selection differential will increase, then the response to selection will also increase or genetic gain will also increase. This is the fundamental concept in quantitative genetics. Here, the heritability in narrow sense means it is calculated from the segregating population, and it is calculated by 1 by 2 D divided by Vp. Here D is additive variance. D is additive variance and Vp is phenotypic variance. Phenotypic variance. We all know that the additive variance is the component of genetic variance. Is a component of genetic variance. Actually, genetic variance has three components: additive variance, dominance variance, and epistatic variance. And epistatic variance can also further divided into three parts: additive into additive variance, additive into dominance variance, and dominance into dominance variance. And additive variance is always heritable and fixable in nature. Okay, so if we talk about heritability in broad sense, heritability in broad sense. The heritability in broad sense is equal to Vg upon Vp. Here we can see that we using the genotypic variance, which includes all the three types of variance. So heritability in broad sense will always be greater than heritability in narrow sense, and heritability in narrow sense will always provide a reliable result because it is due to additive variance. And additive variance is always heritable and fixable in nature. But in case of genetic variance, we can see that dominance variance and additive into dominance epistatic variance and dominance into dominance variance is not fixable in nature. It is heritable in nature, but it is not fixable in nature. Okay, so heritability in narrow sense provide more reliable result than heritability in broad sense. Okay, the breeder equation sometimes also used for a formula of genetic advance. Genetic advance equal to heritability in broad sense. Heritability in broad sense, phenotypic standard deviation and K. K means selection differential. So here genetic advance equal to heritability in broad sense, phenotypic standard deviation and selection differential. This is sometimes also known as Breeder's equation. What is genetic advance? Genetic advance is the improvement in mean genotypic value of selected population over the mean genotypic value of unselected population. Okay, so genetic advance equal to heritability in broad sense and phenotypic standard deviation and selection differences. However, genetic advance can also be calculated from heritability in narrow sense. Okay. Now, heritability in broad sense can be written as Vg upon Vp. Vg means genotypic variance and Vp means phenotypic variance. And this phenotypic standard deviation can be written as under root Vp because standard deviation is the square of variance. Okay. Now, this selection differential can be written as, okay, so here genetic advance can be this, and this is sometimes also known as Breeder's equation. Okay, so what is Breeder's equation? R equal to H and K, and here we can say that genetic advance equal to heritability in broad sense, mean phenotypic standard deviation and K. This is sometimes also known as Breeder's equation. So I think you have got the point. If you like the video, please share it with your friends and colleagues. If you are new viewer, please subscribe the channel.